right, let's get it started. Good morning. So we will, as I said, the room we use Tuesday a little bit small, and so we decide to move to 113. So it's just a lecture hall, lecture room next to us. That's for Tuesday. Every Thursday we will meet here, okay? So I suppose to provide a very relaxed environment here to lead you go through this essential activities which you are supposed to get involved in analyzing single-cell data. And when the big semester for me, there are more than 20 people in this room. At that time, it got a little bit crowded, but now this semester, we don't have so many students, so you feel more relaxed, supposed to be. And if you decide to change your computer from one desktop to another, it will be fun because the configuration are supposed to be the same, but sometimes there are missing parts for one computer for some reason. Some program just not working, then you can switch. You're supposed to be logging in any of this using NetID. Any? You're all fine, right? You can log in? No? Oh, that's interesting. Can you use your NetID at TMU? Yeah. So use your NetID at tmu.edu. I'm not against if you decide to use your laptop. It should be fun, but in general, these computers are more powerful, and you can catch up the process. You can log in now? Okay, yeah. Make sure you use your NetID with the domain name. So this first lecture, we just configure your computer and make it ready. So next week you come back if you decide to use the same desktop here, and you shouldn't do that again because they already configured it. Most importantly, because we use MATLAB as a main language, and it's also the computer environment, so we decide we have to install that here. If you use your own laptop, you should have an on-campus license for students. You should have that automatically, and if you follow the instruction you can find online, you should be able to install on your laptop. Either you use PC or Mac, you all have that access. Remember, the MATLAB is a powerful scientific computing environment. It's a language, and also it's the whole environment. It's advanced, and it's not free. So we have the privilege on this campus because we are a student and I'm a professor. We have a free license, basically. If you are in the company, you have to pay the license fee. If you are in the Iran, in China, some of part of this countries is not allowed to use this because if you apply this environment using in the military purpose, it's against the law. So for some reason, okay? So there's the values of this intelligent asset. So when you log in, let's check if you have MATLAB installed. You can have the search, and because I had that installed previous semesters, so I have that in there as an app. You should click on and bring it up. If you don't, you have to install from ZenWorks. So if you click this icon, in my case, it's dark gray application. then works. So this then works. You through this, you type MATLAB. You have this icon, and you double click. It should automatically install this on the computer here. This process will take 20 minutes, if not longer. So if you don't have MATLAB installed on your computer, you can either switch to the next computer to you. There's high chance that someone already installed that already. Otherwise, you have to install from here. Yeah. Yeah, MATLAB Online is also another option. There's some limitation because that's online system. You cannot read your local file sometimes. But in general, if you decide to use online version, you use registration and use some email address, and MATLAB Online is MathWorks, the company provide this environment. You don't have installation at all. Yeah, just spend 5 minutes, just get yourself ready and set it up. So I will work around and see, okay, we're ready to go, then we move forward. Do you have, yeah, that's good. You have an older version, but it's okay. Yeah, it's very similar.
Okay.
Yeah, but if you type MATLAB here, you should have a new version. It's not here. Can you search MATLAB here and see, do you have any other version? Oh, okay, it's fine. So if you have older version, like 2021, it's still the same. Yeah, you have the latest version. There. So we are good. Yeah, everybody fine. You are not yet. Okay. So we start from MATLAB, but in the same time, we will. I will also show you how to install R package, because some of this cross-validation we want to see. Okay, in MATLAB you get these results. How about in R? In general, other people maybe use R and repeat. And we also install Python, okay? We will try everything we can. So we keep ourselves very, very flexible. So if you want to install R, this will be the R Studio. If you search R Studio and you look for this, the blue circle icon here, and this, if you double click, it will install R package and also the R Studio. This is the running environment which allow you to program in there. So I suggest why not we just double click this and install at the same time. So we have both. I probably already have RStudio here. So in my case, I already have that installed. Yeah, I decided to upgrade my version, so it won't take too long. So what I did is just double click and just wait. You don't click many, many times. The program got confused. You just double click twice maybe. Then it's progress bar, just move forward and try to install. Okay, so in MATLAB, you may already know this, but I just very quickly go through the layout of this environment. So the first time, if you have never used this environment, there's the link that is called Get Started. This is a really good place if you have never used this environment before and you've spent some time to look at the tutorial and see Get Started over there. This is a commercialized package, so all the resources will documented, will prepared. Okay. Want to make the font size bigger so you should be able to see it very clearly, right? Okay, so similar to many this programming environment, you can just start to use this as a calculator, okay? You want to calculate some typical these equations, then you should be able to get the number. So in this way, you communicate with computer here, okay? So I had R installed at the back end also. Well, in here, you have this command window, so that's where you type your command, and then sometimes is this direct calculation of this equation. Sometimes you will see A equals 2, so you assign a variable value to A. Then when you A do plus 3, then it give you 5 because you let A to be 2. So in this way, you interact with this computer, and so you can also call some function. So for example, their function to generate a random number. We will generate 10 random numbers, okay? One means a list, okay? 10, a vector, 10 by 1. So this random number is randomly distributed between 0 and 1, okay? So that's the way you communicate with the program. So if you do generate random number 1 to 10, this give you a horizontal vector, 1 row and 10 columns. And because it's too long, so you have to print out and return to the next line, next row, okay? So that's the command line window. Then on your left top, there's current folder. That's where the file is saved, your current working folder. So in this case, I have my name and the documents MATLAB. So I'm in this folder, current folder. If you do the PWD, this should give you the current folder where you are working in, which folder you are right now. My folder is empty, so I have nothing to show you here. So you have the same place. Then here you have this small window called workspace. Workspace is a memory variable. It's showing you what's right now in the memory here. I just defined a small variable called A, and the value is 2. So you can see this variable A is here. But if you restart the machine or you shut down, this memory will be washed out. It will be empty again. So this is a temporary memory-based storage. Well, this is a hard drive, the file you saved. Okay, there's two places. Any questions here? No, okay. If you have R, Python, so they're similar environment, right? Okay, nothing special. Well, so MATLAB is designed for matrix computation. So it's really, really good for matrices operation. So how can you define a matrix? So if you have a capital A, so remember it's a case sensitive. So now your variable name is different from lowercase a. Okay. In this case, if I do squared this bracket, then you can put the numbers in here, 2, 5, and then semicolon 4, 2, 7. So in this case, I defined a matrix, which is 2 rows, 3 columns. Okay, so you see how you indicate the change of the different rows. That's what the standard data type in MATLAB. MATLAB hold matrix as its basic data type. In the single cell data analysis, you will deal with something like A. Okay, I will not call A. Normally I call big X. It's my input data matrix. The rows can be genes. So I have 2 genes here and 3 cells. And the first gene expressed at level 1 in the first cell. The first gene expressed level is 2 in the second cell, so something like that. It's a very small toy example, but in general, we will have the same idea, even though you have hundreds, thousands of cells, genes, the same thing, right? So this is the one. So let's just call it X. So I assign A to X. Now you have lowercase. This is called a scalar, the single set number, okay, scalar. And you have a matrix A and its copy X. They're the same. So MATLAB is based on the operation to the matrix. So if you say X minus 1, this gives you the whole matrix operation. So every element in the X will compute each element by element, and then everything get minus 1. So you get the results. So you take off 1 value from all the values in there, okay? You don't have to retype it. You just use the arrow key up, down to visit your previous comments, so you don't have to retype. And I will assign this back to itself. So this operation will take a big X and minus 1 from each element and assign back to X. So now X changed its values. Now X is different from original copy of A, right? They're different now. if I want to extract a value from this x, then I give the index of coordinates of x. So this asks, can you return the value from the row 2 and the column 3? So that will, this expression will return 6. So if I want to check what's the expression value of my gene number 2 in cell number 3, so I type this in. It will immediately tell me the level should be 6. All right? So if I do 2, 2, so what's the value of this? This should be 1, yes. But if I do 3, 2, this will give you error message because you index out of the range. You only have 2 genes in your input data. So this will tell you we don't have that number. Your index is out of range. So what if I want to extract the information for the whole gene, gene 1, all the cells value? So I extract the first row. I will just need to see the fourth row, okay? And then you use this. The whole, this indicates I will not explicitly write the numbers of index, but just give me the whole row. And you can do the same thing if you put this one here and the first one there, it will give you the first column. Okay? So this is the indexing system for MATLAB. So this will be the third column of the matrix. Okay? So if I do this, it will return the whole thing. Make sense? So remember, this will be our starting point for the data analysis. But before we get to this expression matrix, there are previous steps. Remember, in a single cell, you prepare the cell suspensions and send to the controller to prepare droplets. And the droplets become the sequencing library. You send it to the sequencing sequencer, Illumina sequencer normally. And that sequencer will give you like FASTQ files. FASTQ files are the raw data you get from the read from the mRNAs. But this FASTQ file will not give you the expression matrix directly. So we put this into Cell Ranger. This is also a program which 10x Genomics provided. Allow you to translate. Okay. So you gave the reference genome, FASTQ, and the reference genome. And the annotation. With those, you generate this matrix. So what I'm talking about in this language here, this one, it's not the very beginning. It's already processed. If you have 2 samples, you will have 2 matrices. If you have 3 samples, each sample generates 1 matrix. So this is one of the samples matrix. Okay? You remember where you are in which step. We will spend some time to revisit this step. If you have this, how can you do this and do that? Remember, this is just one company's solution. 10x Genomics provide this. The other, if they have competitors, they will have a similar pipelines, but different names maybe, okay? But eventually you get the expression matrix and then you can handle it here once you put that in there, okay? Yeah, I just show you a few more indexing, the way you can extract data from this. So another way you can do it is, so let's see, for the rows, we want to have one and 2, okay? For the columns, we want to have 2 and 3. Well, this defined, I don't know if you can see, it's okay from the back, you can see this. So I will start from row 1 and 2, and then 2 and 3. So this will extract this square. So this will return part of this matrix. Make sense? Okay. Why do we want to do this? So sometimes I want to extract cell, which I know 2 and 3 are different cell type compared to cell 1. So I want to do the cell type specific analysis. So that's why I extract this type of cells only and make a sub matrix and work from there. So everything downstream will become cell type specific if I know cell 2 and 3 are different cell types compared to cell 1. All right. So this is just a way you can manipulate your data and see, okay, I want to assign this into X2. So this is my cell type 2 data. My cell type 1 will be the first cell. There's only one cell in the cell type 1, okay? So I just very quickly use this last 10 minutes, just explain to you what's the desktop basics, what is matrices array. So array just, if you have a matrix, is either row or columns is 1, then you have a list of values that become array or vector. So they are same. Then how to do the array indexing. So I just quickly show you some more basic. But those are the tutorials you can visit after after class, okay? Any questions? I also mentioned this workspace variables. That's a place where it shows you what is the variable, the name, and show some information about the variables. I want to quite quickly mention the text. So how MATLAB deal with the strings. So that's how you gave a name of the gene. So this G1 variable, G1, have a gene name, okay? The double quote around it. Then you can have a list of gene. So when we say array is a vector, contains a list of values. Well, in this particular array G, there are strings. So there's string array. So I assign 2 genes name into this array. So I have a list of genes. And this will be the information that I will use as annotation for my X. X contains 2 genes, right? Then I have gene 1 named gene A, and then gene 2 named gene B. So that's all the essential information we need to describe an experiment output from this is a lowercase, but I need uppercase. So this is my data. The most basic data structure I have in order to perform a single-cell data analysis. Okay, from now on, I will call the expression matrix as X. It's a matrix hold integers greater than the non-negative integers, so you have 0 and anything above. And then I have a list of genes with a string array. The number of genes will be equal to the rows of X. So each row has a name for that gene. That's all we need, okay? When I mention next time big X, you know I'm talking about expression matrix for one sample. When I see an array of string G, that's the name of genes. Okay? Now we are ready to play with real data. I will show you some real data. Let's see where we can get. Okay? Questions? No problem. Good. Okay, before we move to real examples, we need to install some packages, okay, or functions which designed for single-cell data analysis. If you don't do anything here, we don't have those functions to help you to handle these things. I just showed you this function. So random number generator. Remember this? If I generated 5 random numbers, this function is provided by a collection of function group. It's called a toolbox. If you see, okay, which random. So this is a built-in function. MATLAB has already have by itself. Okay, because it's such common function, a lot of programs need to use this built-in function. So that's why MATLAB provide random generator as its built-in function. So that's where you search, so where this function is located, and give you the capacity to generate random number. Well, there's another random number generator, random n, if you do 1 and 5. Random n is also a random number generator, but it's not a generate uniformly distributed random number. N stands for the normal distribution, so that's you get values from this bear curve, and that's why it's centered around 0, and you have negative, positive values. Okay, so this function is different from random. So this is a uniform random number generator, generate number from 0 to 1, and equal frequent between all the regions across that range. And in this case, I check which random n the number is also provided by. Well, some other functions more complicated. So like ANOVA test. In this case, it's not a built-in function. You have to install so-called toolbox. That's like external supporting function package. In this case, it's a statistic toolbox, and under the hypothesis ANOVA. So some specific functions you have to use external help to have install those packages. Simple ones MATLAB provided. You want to extend the capacity, so you have to install other toolbox. There are many, many toolboxes provided. The best place to look at, if you know the toolbox name, in this case is the state, and you can use the DOC, the documentation, and it will bring you to this toolbox, and you have the information about what functionality, what extended functions has been provided by this toolbox. Yeah, for example, PCA is one of the functions provided by statistic toolbox. If you don't have the toolbox installed, if you do this, which PCA, it will tell you nothing found. I want you to try whether you have this PCA, whether you have this statistic toolbox installed already. Let me know. It's essential. We rely on this, a lot of this function in there, indispensable. So just make sure you have this installed. If you just type which PCA, if you show it, then you are ready. This is especially true if you install MATLAB on your own laptop. By default, maybe this package, this toolbox has not been installed, although you have a license to use it. So you have to take extra efforts to install that toolbox to make sure those functions become accessible to you. Okay? So how can you install toolbox? If you look at this big icon here, so this is called add-ons. I open this add-ons, and then if you are working on the automated vehicle, you want to use this toolbox, but we are not doing this here. So there are others. If you work on the 5G signal, those one simulations, then you install 5G toolbox. There are many others. Those provided by the MathWorks, which the company produced MATLAB itself. So you have communication toolbox, you have computer vision toolbox. They all installed the MATLAB itself. But many of those functions, we are not going to use them because we are not designed antenna. We're not doing this. Sometimes the function in the bioinformatics toolbox will be useful for us, but we are not rely on it. So if you don't have this installed, it's fine. We only need the statistic toolbox. There are deep learning toolbox. They're for the financials, for the economics. Their image analysis toolbox. So each of these toolboxes contains hundreds of functions which are relevant to this particular application area. Mapping, okay, you do a lot of this. GIS system. If you scroll down, load more. This is the one I suggest everybody should have that installed. So this is statistics toolbox. There are so many essential functions we rely on, so we have to have this, especially in your laptop installation at home. Okay, ready to move on? So you can clean your command window by CLC clean if you don't want those messy Remember the comments, CLC, clean your comments. You clear variable, you clear, so you see you have a lot of temporary variable in your workspace. You can clean them, okay? They all overstart, they not come back. I want you to get familiar with to visit these folders. So now we under documents, if you click this, you come back to the folder which above one level. And then you click this, you see the folder under your username there, desktop, documents, downloads. I have nothing in downloads, so feel free to navigate this, because sometimes you download your file from some website and they are in the download folder, downloads. You need to know how I can navigate and find the file, right? So make yourself familiar with this. And your current working folder is always shown here. And you can use the pwd function to get where you are right now. You can also right-click here to see I want to show this folder of downloads in the Windows Explorer. So you come to this folder here, and then inside it's also empty. If I create a document, see, a test. So I create an empty folder. The problem here is the Windows system is designed not for programming. So the extension of this file, which is supposed to be .txt, has been hid because no this function, this file type. So we want to, as a program, we want to show this so we know what the extension of this file is. So I encourage you to visit the folder here, folder option. So if you do this folder file option, you can get access to this through the control panel. You know how to go to here, and then when you do the view, uncheck this. We need to know exactly what's extension of each individual files on your folder, in your folder, on your hard drive. We cannot let the system to hide it, because as a program, you need to target that particular file, okay? So the first thing you need to do is uncheck this hide extensions for known file types and apply this. Now, the file name has been shown this as a .txt, okay? I have a control of what the extension should be, and I can put in the TAB or become others. So this is essential. So let's navigate back to this default working folder. It's under your username in the documents, that MATLAB. So nothing here, but make sure you are in this folder. Because we are going to install single cell analysis toolbox under this folder. So every week we will use this folder again and again. That's our working folder. And you will install and reinstall, update the package, everything under this folder. Okay? So if you do pwd, you know where you are. Make sure you are here. The toolbox we are going to install is called Single Cell Gene Expression Analysis Toolbox. And on this page, there's a quick installation. That's the code you want to use. To search and find this page and just copy this. I copied this. So this piece of code is just like you type in the command window in MATLAB, but it's just specially designed to visit this web page with my GitHub page to download this package and then unzip into your current folder. That's all. If you have already had the current folder installed from previous installation, it will overwrite and use the latest version. So we can repeat this many times. Every week when you come back here and you update, because I may have previous updates. So you copy this piece of code and paste into MATLAB. So I select copy and make sure I'm in the right working folder, because my toolbox will be installed here. Then I Control V to paste this. The process is automatically download, unzip, and add this folder in. Then we are done. That's all. Yeah. Question.
Is there a difference between the code on your GitHub versus the code that's provided on MATLAB's website? Because the MATLAB code on the website is just unzip your GitHub directory and then add the path for.
They are same. Yeah, they are same. I just add some annotation, so installing, so how long it take, whether. Yeah. So in the GitHub, we have a simplified version of this, but it just unzip this, add the same thing. The most essential one are these two. Yeah. There are different ways to install this, but this will be the most straightforward, and make sure you have the latest update. If I make any change here, then you do this again, you immediately have that update. So this toolbox is completely under our control. If you say, okay, there's something wrong, let me know, I fix that immediately for you. If you want to say, I want this function to do this, to me, maybe just take a little bit of tweak, and you get the function we need, okay? So this is very different from we use other people's package, and they change something we don't know, like our reason have a huge upgrade. Some previous programmers is crazy. My code, it was working, but now stop working anymore. But this, I have a, where is the author? Okay. With this, installed, let's see what I can do. If you don't install this, you don't have this function. So this function just helps you to simulate a matrix. Okay, you don't have to type that in this, let the program according to some rule and our understanding of the distribution of data generate this simulation, okay? So how can you do the G? G will be plus 1, 2, string 1 to 2, because we only have 2 genes, right? So this will help you to create a name of the string of this. We don't have a real data, but we just call it gene 1, gene 2, gene 2. In this way, you define a string array, and you simulate a matrix, okay? So we can take a break, 5 minutes break, and come back. We will simulate a huge expression matrix, contains maybe 200 or 500 genes, and then see what we can do next, okay? 5 minutes break. Okay, remember this is the most fundamental data structure we need. We need what? Big X, small g. Big X is your expression matrix, and small g is the name of genes. Okay, so if anyone asks you to do single-cell data analysis, let him or her to give you the big X and the small g. That's all we need for the downstream. Okay. So where can we get the data? Okay, how can we get, for example, publish the data? And the most common place you can have, I don't know who have used this website before. Anyone? Some of this okay. So this will, when you have a gene expression analysis, you want to publish a paper, normally the editor will ask you for accession numbers, and you have to upload your data, mostly expression matrix, to the geodatabase held by the NIH, especially if you have funding from the NIH itself is compulsory, so mandatory. So from here, for example, we can search. Normally we will have a shortcut because the technique we use is 10x. So I put 10x something, and then, for example, genomics, single-cell RNA-seq, lung cancer, and then with these combinations of keywords, I can have some like keywords level filters. Technology we use from 10x genomics, single-cell RNA-seq, and the data set is relevant to the lung cancer. Mostly, I should get the data we need. There are 103 results here. And then I will read through the annotation. So as an example, so this data is published 2 days ago. Then it's a cancer cell line study, and there are samples treated with this compound. I'm pretty sure they have data from before and after treatment. So look at their data. You see there are 4 samples. DMSO definitely will be the control that you add that in. Then they probably did 2 biological replicates, 2 control, maybe different cell lines, maybe. Then if you click inside one of the samples, and this is the matrix they provide. Okay, they provide the R-based, the RDS, which they process with R. So that's the way you found the expression matrix provided by the published paper. Okay, in the paper they normally will give you the accession number, which is this SCE, etc., etc. You can follow that, come to this page, and find their data and download this matrix they provided. Let's check another. So this is a mouse study. Six samples in human, so I visit this page. They have a PBMC, which is a blood samples. Then if you click inside, you see this very typical, the one we just saw before, RDS is not a typical upload. They're not the best practice. This is the best practice because the cell ranger, the program I just showed you here, will transform the raw data into something like this. Okay? So this group of investigators did the right thing. They upload the raw matrix. This is the matrix file. It will contain the big X. This called feature will contain the information for the small g, or the name of genes. This is barcode is for the cell name. So it's a random barcode, 10x genomics, used to differentiate one cell from another. Normally, this is not essential, but we need these two files. Okay. Questions? So if you indeed, the investigator, they upload the RDS file that's prepared using R, we have a way to download the R file and convert into this. It's not very difficult for us. Okay. So normally you can download these files each and put them into a folder. Let me do it. So I can download this file, and this file. The matrix file typically larger than the rest. Okay? It'll take some time. So in my download folder, I have 4 files, and this one was the one I just created as an example, so it's useless for me, right? So I just delete it. So I have 3. So I can see, put a example. I put these 3 inside. Remember, as I said, you need to know the extension. So those files supposed to be, after the unzip, should be MTX, TSV files. But right now, because they are zipped, you don't see them. Okay. Now the extension is GZ. We can take a look. So I'm interested in first the feature. So I can do right click and extract here. Now, this particular file has been unzipped as a GT. Open with. So I open one of the feature file I download after unzip. I can open it with a text editor here using Notepad. I see several columns of data. This is a ensemble systemic gene name, and here are the gene name symbols. And then this column just tell, okay, this sample is from gene expression. This is completely redundant. So the information we really need is the second column, right? I don't know if you have the same. If you use different examples, you may have different name of genes, depends on what you are. I can tell this is a human sample because all the gene name is capitalized. If you have mouse genes, they will capital the first character in the gene name. So that will be, for this particular gene of mouse version, it will be capital A, small g, R, N. Okay, by looking at the name of gene, I can know this is a human sample. Because when they doing this mapping, they map to the human reference genome and using the human gene annotation. All the information of human gene name has been carried on and put in here. If they map the sample into mouse, human mouse so close, so they still will get the matrix. They will have a name for the mouse gene names. You will get the matrix as well. All right. Now I take some time to extract this MTX file. So as I said, this is a matrix. So we are going to use this to read into the memory of the computer as a big X. And if you open it, so the extension of this called MTX is a special format to save matrix data. I don't even have a correct. So let me open this again and drag matrix into here. So I open this MTX file. The format is called matrix market, matrix coordinates. This file, this version. Okay, these are the index, the position. Maybe it's a row 4346, and the first column, and the value is 1. So this is a huge matrix file. Okay? So that's where the information we will read in to reconstruct our huge matrix X in the MATLAB. So those are the index. So we know how to do the index, this position I, J, and value 1. So we can prepare the huge matrix with all zeros and fill that in and reconstruct that in memory. Any questions? We are not going to deal with raw data because I have a function already written to help you to drag that information input into X and G. Remember, X and G are our essential data type. We don't even have to unzip them. The function we developed automatically unzip this and read the information and put it back. So you don't have to worry about those steps. So I can delete this, delete that. That's a 3-row data file I download from the website. So I need to remember where I saved the file. I saved into the downloads folder example single-cell RNA-seq data. Come back to here. So I'm going to read X and G. So this function you can use to read the folder, because there are 3 files. We don't have to say, okay, so file 1, 2, 3. They all standardized named. So I only need to know the folder name. So the folder name is this, right? So I copy the folder name and gave to the program. Let's do something else. Folder name. Yeah, so I assign the folder, the full file, the folder name into a folder name variable, and I check this folder is there. Yes, it is a folder. So answer is 7. If it not exists, it will return 0. So I know I'm doing the right thing. So I found the right folder. Then you need to give the folder name to the program. I put a semicolon at the end because otherwise the content of the G and X will print out on the screen. Will very, very messy because it's a huge data set. And this will surprise the display of those feedback information, but only do the assignment to the level, to the variable values quietly, okay? So that's important to put a semicolon at the end. Without this feedback. Okay, this function, smart enough, will go to that folder to unzip them first, and then combine the information of the 3. The variable X and the G right now in the workspace, they are small examples. There are only 2 genes, right? This is random generated. But after this, these 2 should be overwritten, and you should have a real data from that particular sample. Yeah, we just have to wait a little bit. The information from a barcode will not be used unless you ask for the barcode, because right now we only ask this function to return 2 variables. If you need the barcoding system, you can put comma B, and then the barcode system will come back. So I don't know how long it will take on your computer here. It shows me might have a big X matrix already. The contains 26,000 genes and 14,000 more cells. So this is the dimension of your matrix. And this is the gene, okay? Have you gathered the same thing you are working on? Your computer may be faster than what I have here. You can double-click G to look at the name of genes. The first G is microRNA gene, second, and then there's some unnamed genes, the long non-coding RNA. Okay? And the big X, if you double click, you won't be able to see the values because the big X is stored in a compressed way to save memory space. It's called a sparse matrix because this matrix contains a lot of zeros. If there are zeros, I will not fill that in to occupy the space. I only encode the information that will with non-zero values, so to save much space. But you can force the program to give you the full X. So if you do X, so this will convert this sparse matrix into a full version. Because for demonstration purpose, I want to show you the values. So if you double click X, you will get the values in there. So this is a function. Remember to put a semicolon. You can try without a semicolon. It will take some time to show the values on the screen. Mm-hmm. Yeah, I think so. Yeah, I think it's just slow.
Oh, okay.
Yeah,
So,
no problem.
Otherwise, like it just takes time.
Yeah,
Okay.
it's a huge matrix.
Right.
It takes some time.
But my folder was like this. Like this.
Yeah.
That's fine, right?
No problem.
Okay. Right,
Mhm.
okay. Thank you.
Mm-hmm. Running. Yeah, so you use Control C to stop it. Yeah. Yeah, that's why you put a semicolon to stop the printing on the screen. Yeah. Any questions? Are you okay? So now you can double click in the workspace and take a look what's in the X. This is real data we download. You're supposed to see a lot of zeros. Okay. That's why people say, okay, analyze single cell data is difficult because it's such a sparse matrix. What can we do with all the zeros? There are only many genes, maybe only 10% of non-zeros, right? Yeah, so we don't have to deal with this problem today because we have a special time to discuss the QC, okay? How can you do the filter? Basically, we want to condense this huge matrix into relatively small ones to throw away a lot of genes. throw away some of the low quality cells, because if they are too empty, they're sometimes important, but in most of the case, they're not provide enough information for you to do the analysis. Okay, in 26,000 human genes, we probably will throw away a lot of these microRNAs, a lot of non-coding RNA, because they are not poly A-tailed, supposed not to be enriched by the 10x genomics. Remember, the barcode contains the poly T-tails. It should be able to amplify the information from a poly A transcripts. We want to remove some genes which expression level is so low. Okay. So we have to do some statistics with X compute. See, for this gene, let's just try maybe gene 2, see how many zeros in there, the percentage. So how can we compute? And then we decide whether gene 2 is important or not. Okay, so let's just do that. Do you know how to extract the gene 2's expression? Gene 2 in the second row, right? So the indexing will be the 2, the whole row. That's the gene 2. This is X2, right? So I just quickly extract gene number 2. What's the name of it? This gene, G number 2. So we are dealing with this gene, and let's check what's the gene is for. So this is well, this is RNA gene, so this is not a protein coding gene in human. So we are playing with the data from this particular gene, and we have its expression level extracted from 14,188 cells. So this X2. Look at this dimension is 1 by 14,188, right? So double click this. So what you can do with this X2? I can first see average expression, right? So I can do sum X2 divided by length X2. This is the average. So what this tells you, or you can use the mean X2. So this is average expression. So this gene is completely empty. All the cells in this particular sample are 0. Unless you have another sample you want to compare. So maybe you have this sample and another sample where you look at the same gene, and in another sample, the average is really high, then you don't want to throw this away. But by looking at this particular sample alone, I believe this particular gene is useless. It's empty gene. So I should remove this gene from the downstream analysis if I focus only on this sample. Okay? So you can try another gene, for example. I still call it G2X2 just for the convenience. And the name of this gene is 231. Okay, it's a long non-coding RNA genes. Then I can do another test. Well, we have another empty genes. Okay? So although this is like a monster, but we know it's so empty, some of genes will be removed. Then we will do some analysis. After remove gene, we will look at it by columns. Columns are the cells. Some cells, we just not sampled enough. The overall expression, if you sum the whole column, again, some will be really low. We will throw those genes away. So eventually, you will end up with a genes matrix, about 10,000 genes, and normally will be 10,000 or 11,000 cells. So that's a typical, the size, the dimension of the matrix we will get for the next step of analysis. All right? So let me look through the G, and do you have any idea which gene is supposed to be housekeeping genes and you believe should be highly expressed? Because we tried 2 genes randomly. One is number 2 genes, another 231. They're all empty. And which, if you look at it by name, do you recognize any gene that you believe should have a non-zero? Otherwise, the cell should not survive. That's the definition of the housekeeping gene, right? So I just randomly browse through the name of genes. What do you think about this transmembrane protein? Let's check the name. This definitely is a protein coding gene. So I have some feeling this gene should express at a certain level. So I look at the index. The index of this gene is 722. So now instead of 722, the gene name 722 just confirm is this transmembrane protein. And then look at its expression. I start to see some non-zeros. So at least in the cell number 4,875, this particular gene expressed at a level 1. One copy has been detected. So if I do average of this, it's pretty low, but now at least it's not zero. try another one. Do you have any idea which gene I should look for? Actin? Beta actin. What's the name of gene? The ACT, something like that? Because they all are official names, so ACT. So I do search ACTB, right? Found. I just try to see whether, so the gene number is this. So I can see, okay, my X2 get this number, and then that's the expression of this gene. Is that okay? So we see this non-zero across all the cells almost. So definitely you found a housekeeping gene by name, and the expression level confirmed this is a housekeeping gene. Is it good? So what you can do is you do a plot X2. If you interested in what it looks like of this particular gene's expression across those files. Normally, plot a file, you need a curve, you need an x and a y axis. But in this case, we only provide the value of y. And this plot automatically gave each value an index from 1 to the end. So you plot 1, 2, 3, 4, 5, and then the value of the inside the X2. The housekeeping gene, you have expression level average maybe 150 somewhere. But some cells have a high expression of this gene to the really almost over 1,000. But on average, you have this. Maybe a few zeros we cannot see from the plot. So this gives you the profile of gene expression of this particular given gene, housekeeping gene, in this cell population. Okay? So this plot is useful for you to understand the average expression of a gene and also the variability across individual cells. Okay? I think today we already go through quite a lot of information, right? You prepare your working station and rename the file by showing the extension and know where to download the raw data and how to install the package, bring up the essential function. You can get X and G. All right. I think that's more than enough for today. And you show the genes expression for this. I think the last thing I want to show you is once you have this, you have X and G, what you can do is use this toolbox, GA2, you put X and G as an input variable. To create, let's see, we do this. Use tab if you, also the case sensitive, so you have to type in the same way. Use the tab to automatically complete the coding, and then you need a G and X, these two. So this create a compound variable, mix X and G together. So otherwise, we have to deal X, G separately. But now we combine them, make a combined object. It's called a single cell experiment. So all the information will be held into this new variable called SCE. So SCE, if you have that, it should contain X and G, so you don't have to deal X, G separately. We will focus on SCE for all the whole semester, all the downstream analysis, okay? With SCE, you can do the visualization now. So after bringing this up, I show you, or you can do this. We will finish today's lecture and come back for using the whole semester to go through some of these major functions in there. Now, you read this matrix information and the gene information into interface like this, okay? Each point is a cell. It has been randomly embedded into this 3D space. We are not doing anything yet. We are going to cluster them, remove their location later based on the similarity between cells. But for now, they are just randomly in there. You can select some genes using the first button here. Select a gene ACTB2. You select this gene. You can look at this gene's expression in these cells by color code them. For now, so that's all we need to know this week. Yeah, it's a slow computer. Okay, there a row of this small icon here, okay? If you highlight one of these, rotate 3D. So after you active this, you can start to rotate. You can zoom in. And zoom out and move, okay? You can then later you brush to select cells to do something later, okay? You can select some cells. You can save this picture. And later, if there are information, you can use this data tips to show that information. But right now, there's nothing you can do with this, okay? Okay, so this is all for this week's lecture, computer session, and let me know if you have any questions. Otherwise, I see you next Tuesday in 113. Okay?